Hello, everyone. Welcome to today's episode. My name is Sadi Anwar, Acquisition Editor at River Publishers. Today, we are excited to welcome Carol A. Rai, founder of ImmutriX Therapeutics Inc., and a leader in medical technology innovation. With over 40 patents and a strong track record in biotech and policy, Carol brings incredible insights to this episode. We are also joined by Professor Pranela Rameshwar, Editor in Chief of International Journal of Translational Science. With that, I would kindly ask Professor Pranela to take the lead from here.
Thank you. Thank you, Sadia. So, Carol, it's so good to see you again. This podcast is stemmed from a long research that ended up leading to the most recent publication to you. And you knew I was working with exosomes. And so you contacted me to ask if the carbon was capturing those exosomes. Maybe if you can tell the audience just a brief your interest in this and cancer.
Yes, well, it was an idea that I wanted to pursue because my daughter had been diagnosed with glioblastoma, and being a very independent young adult, she chose not to do radiation or chemotherapy for her treatment. And so we basically did a bit of a global search to find a method of treatment that was something other than the standard of care. And the result of what we did, we at the time went first to Karolinska in Stockholm, and then we ultimately went to Germany, where she was treated using an apheresis and plasma exchange process. And it was then that we observed that many things, but the treatment definitely contributed to her well-being, but that other patients also having that experience, when the treatment stopped, they basically reverted back to their condition. And it's a long story, but I ultimately met people that suggested that we look into using adsorbent materials that had already been tested primarily in the Ukraine and other places, had a long history. And one thing led to another. And ultimately, we developed a manufacturing method to using synthetic materials and carbonized materials that had consistency and of both size of the carbon beads, porosity of the carbon beads, and importantly, the overall uniformity and whole blood compatibility. And so we thought that that was a good approach for a number of things, but I didn't have a clue how it was going to work in a cancer situation. And when I contacted Pranela, she really introduced me to the concept of exosomes being the carriers of cells that are typically not under the surveillance of chemoradiation, and that those carriers would protect the cancer cells in circulation. And ultimately, and over time, those cells could be released back into circulation, whereby metastatic disease would develop. Am I right, Pranela? And so that intrigued me. because we had done enough research to know that most cancer deaths, or many cancer deaths, are result in a metastatic state. So when I contacted Pranela at Rutgers University, her students undertook the benchtop studies to determine whether or not our carbon in our system would in fact remove exosomes that we knew to be the carriers of these cells. And that's how it all started.
The reason this journal is so good for translation is that it opened an idea that what Carol developed to the whole area of cancer. So, but cancer people may not work on this, but other people are working it to understand how these exosomes might be bind to this carbon, because the pores are small, but the carbon is in. So I wonder how this was happening, but the exosomes can put itself into cup shape and could be sitting in those pores. But what was intriguing, what we found, is this certain carbon was specific for the cancer exosome and not the healthy exosome. So we wonder if there's something on that exosomes that's attracting it to the carbon. And so what I foresee in the future is to get those exosomes back from the carbon and then do some proteomics and to see what is outside of that carbon. And whatever is outside of that carbon, then we can maybe attach that to something else, because I will let Carol explain that, because you can attach it to a carbon and maybe make it more efficient in gobbling up those exosomes on those cancer patients. How do you foresee
Yes.
that, Carol?
Well, Pranela, we have begun mapping epitopes for all kinds of conditions and diseases, and we now believe that we can more efficiently and bind specific targets of interest, because when we did the earlier work with you, we were basing everything upon the size range of the molecule we were trying to bind to the carbon. We now believe and are working to functionalize the carbon to very specific targets. Our early work in that regard was completed last April. We'll be continuing that work with these epitopes and other specific binding sites that can be attached to the carbon. We know now we can do that. With that advance of the carbon, we'll get two benefits. The porosity of the carbon still is, we don't want to alter that so much, but on the surface of the carbon be able to also bind these other interests. So the work will be resuming in September. We have a laboratory that's dedicated to this project, as well as some others. We will be looking at some orphan disease epitopes that we can bind to the carbon as well, believing that this may be a way to enhance the mitigation of those conditions due to the molecules in circulation that are contrary to health. And if we know specifically what we're targeting, we believe we can effectively cause a binding affinity to the carbon.
We did not just find different carbon bind to exosomes from ovarian cancer, different carbon bind to breast cancer, triple negative breast cancer, and different carbon to HER2 positive. So I wonder if
That's
you
right.
foresee something personalized, something, if we can put it analogous to CAR T therapy. So if we can do a proteomics or a lipidomics, take the exosomes, once they bind to the carbon, see what's unique about this compared to the ones that didn't bind. And so you have that as a personalized medicine. You have that epitope attached to the carbon
Absolutely.
and available. So if a patient have ovarian cancer or glioblastoma or maybe breast cancer, because those are the ones we studied, and then they see that particular epitope on that cancer, then aha, you have available on the shelf that epitope with that
Yeah.
carbon that could be, you see that you force it, do you envision that could be a future treatment?
Absolutely. I am convinced that we would not be here today talking about this if we weren't hadn't had these indications of the unique and uh, binding affinity that the carbons that we make has. We have dozens of, I'll call it recipes, and uh, applying the work with and testing these different carbons is what we believe will lead us to that personalized therapy for binding those molecules out of circulation.
We did not look into the hematological cancer, and there's a possibility that exosomes might be important too, to set up a distant site for those cancer to go. And one of the thing I thought about is a lot of leukemia, dentists actually will diagnose leukemia in the gum and then refer them to the hematologist. But I know now someone at our dental school who is now looking into this. And the question is, where is this cancer coming from? Leukemia started from the gum and then or from the bone marrow, and then maybe exosomes can be setting up because you have a similar microenvironment in the oral cavity as the bone marrow. And so I think going forward, Carol, maybe might want to look into those blood cancers actually also.
Uh, Pranela, I'm committed to continuing to work with you and others to explore these possibilities. I will tell you, at one point, we did a very interesting study in the oral cavities following extraction of teeth, and we created a swab, basically, that contained the carbon beads, and the healing in those cavities and the lack of infection that or the fact that no infection occurred was remarkable. And so we know that anytime there's a bacterial translocation, even in sepsis, there's things that the carbon can mitigate. And the big thing we saw in the treatment in the oral situation was that the healing was so rapid. And so yes, these are all
Yeah,
areas
yeah.
that the materials need to be further explored.
And so, Sadia, this is why this journal is so important, because it captured the early science. And as you listen to Carol, it's exciting because this paper now is available to everyone to look at and see the potential of this technology now.
Yeah, yeah, I totally agree with that.
Mhm.
I just wanted to ask that how you can see the other factors, like the other influential factors that can contribute to those cancers, like environmental factors or maybe heredity.
So that's a good question. So a lot of it is hereditary. If say you have a hereditary cancer, there's not much you could do, but the mechanism as the cancer progress end up to be similar. And then you have additional mutation. And so I think there's something unique that surprised us actually, that different carbon was able to capture those things. So even though you have a hereditary cancer, is it possible that it's expressing, those exosomes are expressing the same molecule? The other thing, those exosomes could also be coming from the microenvironment. And so if you capture those exosomes, then they won't be able to prepare the microenvironment to get the cancer to survive, if it makes any sense. And so this is a whole area, and I think this paper is going to start having someone out there in the world to look at it and said, you know what, there's something here. And we've seen this all the time. Work has been done, and it's being picked up somewhere, and then someone picked it up and end up in an explosion that is good for impact on public health.
Well, Pranela, I'm a firm believer that we've had a lot of divine intervention in these early observations. For instance, with when my daughter was first being treated, it made no sense to me that what was being used had some impact on the improvement in her overall well-being, and that as soon as the treatment stopped, things got worse again. Well, then we went to the next step and started looking at the materials used in those columns that were being treated, and they didn't have they just weren't consistent. They were various, whether they were antibodies, whatever they were using in those columns. And it occurred to my daughter to say, why is everybody making this so difficult? If they can clean up the Gulf of Mexico from an oil spill, why can't you clean up my blood?
And
I
thought
thought about that, and I thought,
and that's what inspired you to start a company? That statement?
That's what started it. And at that point, then we undertook a test. We did this at Texas Tech with Dr. Jan Simone to look at 17 commercially available adsorbent materials. And out of those 17, we narrowed it down to 3, and then we narrowed it down to activated carbon. But the carbon that we had at the time was from an organic source. And again, you didn't have the consistency that would allow you to predict outcomes based upon the studies. I mean, there were too many variables. And that's when we turned to obtaining and then manufacturing, improving the manufacturing process of a synthetic precursor, carbonized, that could be brought to medical grade and have hemo and biocompatibility. So those were the steps that we went through, and that's how we zeroed in on the carbon production being a key component to the development, use and development of products that would treat whole blood for targets of interest.
So, Carol, this paper does not reflect that story behind how this company started. Maybe you might want to write a short commentary or something so we can put it in the journal. What do you think, Sadia?
I think it's a great idea.
Because as I'm listening to Carol, I did not know the whole, the journey. I did not know the journey. And this would be inspiring for any young person who might be wanting to start a company, and you have to have a reason to start a company, and you have to have a passion for it, and the passion was unfortunately stemmed from her daughter's illness. And Carol, how long did your daughter live after she was diagnosed?
She lived 7.5 years.
which is highly
which
unusual.
is
I don't know anyone with
I'm
glioblastoma.
sure it's in the record books. Uh, Jill did not do chemo or radiation, even though that was the standard of care recommended following the surgery that we had in Stockholm at Karolinska. We went to Karolinska because the medical treatments in America and the liability concerns, they just felt like if she wasn't going to follow the standard of care, that it would be not effective. At Karolinska, they did the surgery when others wouldn't, but they still recommended chemo and radiation. And Jill determined that I'm not going to do that. If I'm only going to live weeks or months, I'd like to not be under the effects of those treatments. Yeah. And we got 7.5 years, which is remarkable. She died at the age of 44.
It's an amazing story. It is paper that we now publish of an amazing background that I would like the audience to know the story, what Sadia. And so, because this is amazing in many levels, actually, because there might be a young person who said, have a passion for something I want to start. And Carol put her all into this, and look where it's led to now. This could be huge, actually.
Yeah.
Well, we were hampered by being, number one, a small company, number two, not having sufficient funds to do a lot of things. And then when COVID hit, we had already established a supply chain for our hemoperfusion pump system, our columns, etc., and everything was interrupted. But we did some testing, and we found even the effectiveness for treating the cytokine storm and reducing the chances of the advancing of the COVID thing. And we just did a lot of things. We just didn't have the resources, human or financial, to do everything. And so we turned to, for a focus standpoint of the business, we turned back to what we were already developing in the veterinary market. And I'm very pleased today that we can boast that in treating toxicities, hemolytic anemia in dogs, we've got sepsis studies going on. We've got a number of things with companion animal studies that are being conducted by veterinarians. And we've treated over 1,000 dogs with 100% survival rate. And all of those dogs were brought in to be euthanized.
But if this would work in a dog for this disease, it should be able to work in human, because a lot of time people do experiments in dog to get the approval to work in human. So, Carol, when you write this story, I would like someone to write maybe what the outcome of this dog study also, because this is an impetus to go to human. If you have 100% success for him, for anemia, for sepsis.
Amazing. It's been amazing in treating toxicities. It's just in the veterinary market, when you're in the companion animal space, or actually in the clinics, they treat the dogs, they're up wagging their tail and going home within an hour after treatment, and were brought in comatose. Everybody's cheering and happy, but the data doesn't get reported the way it would need to be for full studies. We're now doing that at many institutions and teaching hospitals in the veterinary space. So I expect a lot of our results in veterinary medicine will translate translate and be a good indication for us to do other conditions. I will tell you that we do have a 510K application in with the FDA that we expect to be approved this year, and that's for treating toxicities. We know in drug overdose situations and other things that it's very effective. It's very effective. We're not competing with Narcan. We love to see that Narcan can get those patients that have overdosed to the hospital, but how do you get what made them overdose in the first place out of the blood? Well, it's our treatment. So those are things that we're going to continue to pursue, because too many young people, old people, everybody are dying of these overdoses, and it's no different with animals, because over-the-counter drugs are very toxic to dogs and even pharmaceuticals. And we have treated just about everything you can imagine in canines, and they've all survived. So translating that for the future of treating humans with the system. Our system is complete. We have a hemoperfusion pump system designed specifically for treating whole blood. We have a column design, a patented column design that makes the treatment of whole blood better suited than older types of columns. And then the material in the column we manufacture, and that's the activated carbon. Now, if we can functionalize that carbon to be even more specific, we think it will have many applications for treatment. And we welcome ideas and opportunities to work on those.
Well, the publication, if anyone read the discussion, we are on the road to this with the functionalized carbon. I think we're on the road. And this is why I'm so glad for people to read this paper that I'm really excited about, actually.
It is so interesting to know about these recent advancements, especially when it comes to the treatment of cancer. And we can, of course, continue, but now I think we should wrap up. And thank you very much for your support and for joining us for this special podcast.
Yeah, thank you, Sadia. Thank you, Carol. Okay, bye.
Thank you, Pranela. Bye-bye.